The identification and characterization of acyl-CoA lysocardiolepin acyltransferase in brain by Ryan Bradley and from the group of Robin Duncan. That's all.
Thank you. Everyone hear me? Okay. The Human Genome Project has allowed us to rapidly identify a large number of protein families through similarities in their genetic sequences. One of these families, the AGPAT family of proteins, has a conserved sequence in all its isoforms, which is a histidine X4, where X is any 4 amino acids aspartic acid sequence. And within the AGPAT family of proteins, this sequence is known as motif 1. What's known about this sequence is that it's thought to be responsible for acyltransferase activity within proteins that function in phospholipid synthesis. AGPATs function in the Kennedy pathway, which is the pathway for triglyceride and phospholipid synthesis. And so an AGPAT family member converts a lysophospholipid into a phospholipid by esterifying fatty acyl-CoA at the SN2 position of the lysophospholipid to convert it into the phospholipid. If a protein is truly functioning as an AGPAT, then it's converting lysophosphatiding acid to phosphatidic acid. And note that there's no polar head group here attached to the phosphate. If there was a polar head group, for example, a choline, making this species lysophosphatidylcholine, and then with our resulting phospholipid being phosphatidylcholine, then this protein is not an AGPAT, it's an LPCAT, a lysophosphatidylcholine acyltransferase. Similarly, if that polar head group was a serine and phosphatidylserine was being produced, this enzyme would be an LPSAT, a lysophosphatidylserine acyltransferase. So far, there have been 11 isoforms of AGPATs that have been identified in both humans and mice. They show a diverse tissue expression, but at the cellular level, they mainly localize to the endoplasmic reticulum. And most do function as AGPATs in that they convert lysophosphatiding acid into phosphatidic acid. They've all been characterized except for AGPAT isoform number 4, which is the basis of my master's thesis work. And so to start the characterization of this isoform, we first wanted to see where it was expressed at a tissue level. And we found that AGPAT4 is predominantly expressed within brain. Here we have different brain regions. We also saw expression in heart and in renal white adipose tissue. After that, we wanted to see where it localized to within the cell. And so we transfected in an AGPAT4 GFP chimeric protein into HEC293 cells. And what you're seeing here is GFP acting as a reporter to where AGPAT4 is localizing within the cell. We counterstained the nuclei of these cells with DAPI and co-transfected in a mitochondrial tracker protein that stained the mitochondria red. When we merge these images together, we can see in yellow here the co-localization between AGPAT4 and the mitochondria. And specifically within this image, we see an 86% localization, which was huge for us because most other AGPATs have gone to the endoplasmic reticulum, but now we're seeing an AGPAT that has a high percentage of co-localization to the mitochondria. We also have this Western blot here of cells that were infected with AGPAT4 adenovirus, which were then fractionated, and we see a high localization to the mitochondria. So we started thinking, okay, well, if it's in the mitochondria, what could it be producing? What type of phospholipid what's so special about the mitochondria? And mitochondria has a phospholipid that's specific to it, which is known as cardiolipin. And cardiolipin is kind of a special phospholipid. It's a diphospholipid, so you can see the first phospholipid here, the second over here, and it's joined together by glycerol. Cardiolipin is predominantly found in the inner mitochondrial membrane, and it's involved in mitochondrial energy metabolism. A lot of proteins that function in the ETC, electron transport chain, need to function in a cardiolipin-rich environment. Cardiolipin is also important in regulating brain function. The peroxidation of cardiolipin can contribute to the development of both Alzheimer's disease and Parkinson's disease. Barth syndrome is a rare genetic disorder in which there's a defect in cardiolipin synthesis through a mutation in the cardiolipin remodeling gene to FASN. And Barth syndrome can lead to impaired cognition later on. And so to test what AGPAT4 was producing, what type of phospholipid it was making, we set up an assay where we had a control group and an AGPAT4 group. And to each of these groups, we added in a radiolabeled fatty acid, radiolabeled oleoCoA, as well as a lysophospholipid. And we can see here just the different phospholipids lysophospholipid that we tried out. We ran this assay and then extracted the lipids and ran that on thin layer chromatography, and then exposed that onto film. and that's what we're seeing in this image. So this is the amount of phospholipid produced and the different species in the AgPac group as compared to the control. When we look at phosphatidic acid, we see that there's no real change of PA made in it in the AgPac group as compared to the control. So AgPac4 isn't functioning as an AgPac. We see that similarly with phosphatidylcholine and phosphatidylethanolamine. We also wanted to see, we also tested out lysophosphatidylserine, but it wasn't enough to see on the film. When we get to this group, however, we see that the amount of phosphatidylglycerol that's synthesized by AgPac4 is significant as compared to the control. When we see the same effect with phosphatidylinositol and cardiolipin. So in vitro, AgPac4 is producing PG, PI, and cardiolipin. But what happens in vitro is not necessarily what happens in vivo. And so AgPac4 is either acting as an ALCAT, an acyl-CoA lysocardiolipin acyltransferase, which would be converting lysocardiolipin into cardiolipin, an LP-GAT, which would be producing phosphatidylglycerol, or an LPI, a lysophosphatidyl'inositol acyltransferase, which would produce phosphatidylinositol. And so to test what it was doing in vivo, we transfected HEC-293 cells with AgPac4 protein and waited 48 hours and then subjected them to fatty acid analysis by gas chromatography. And we found that within the cardiolipin produced in the AgPac4 group as compared to the control group, there was a 62% increase in the number of fatty acids. The number of fatty acids is a surrogate measure for the amount of phospholipid that was produced. And so AgPac4 is synthesizing cardiolipin in vivo. But what about PG and PI? We saw that these were products in vitro. PI had a p-value of 0.2, so it was not deemed to be significant as compared to the control. And PG had a p-value of 0.059, so it was borderline significance, but it was not found, it was on the wrong side of significance. We were also able to tell what type of fatty acid species was present in the AgPac4 cardiolipin. And we found that AgPac4 preferred to acidify saturated fatty acids as compared to MUFAs or PUFAs. And so as exciting as the mitochondrial day was, this was even more exciting because now we have an enzyme that's functioning as an acyl-CoA lysocardiolipin acyltransferase. It's functioning as an ALCAT both in vitro and in vivo. ALCAT1 has already been identified. It was the AgPac formerly known as AgPac8. ALCAT1 is predominantly expressed in the heart, whereas R-ALCAT is predominantly expressed in the brain. ALCAT1 prefers to acidify HUFAs, highly unsaturated fatty acyl-CoAs, which makes the cardiolipin that it produces more peroxidizable, whereas R-ALCAT prefers to acidify saturates, making it less peroxidizable. And so we're renaming AgPac4 ALCAT2. And how we think this may be working is that immature cardiolipin is formed in the Kennedy pathway. And the reason it's immature is because the fatty acid species here at the SM2 position was just kind of slapped on during synthesis, but needs to be specific for the type of tissue that it's in. So if this cardiolipin was going to be in brain, you might expect to see DHA or arachidonic or oleic acid there. If it was in heart, you might see linoleic acid. And so in order for it to become mature cardiolipin and function within the membrane properly, it needs to be remodeled. And this goes through the Lanz remodeling pathway, where a phospholipase A2 will come and cleave off the fatty acid at the SN2 position. And then ALCAT2 can come in and esterify its fatty acyl-CoA, so this would be a saturate there to make mature cardiolipin. This mature cardiolipin can then be formed, or can then be used in the membrane. And so to summarize, we've renamed AgPac4 into ALCAT2. ALCAT2 converts lysocardiolipin into cardiolipin. It's predominantly expressed in brain and shows a very high localization to mitochondria. It also prefers saturated fatty acids. Currently, we're completing the characterization of this enzyme, looking at the biochemical analysis. So we want to find out its optimal pH, its dependency on calcium, and we're generating kinetic curves. We're also really interested in its role within mitochondria. I'd like to thank my supervisor, Dr. Robin Duncan, my lab members, and Dr. Adam Metro from the Stark Lab at the University of Waterloo for his help with GC. I also like that. Thank you.
Thank you, Ryan, for your timing. So we have time for a couple of questions.
Very nice talk. Usually in the Lanz pathway, in terms of phospholipid metabolism, there's an increased complexity. You're taking off a saturated fatty acid and arachidonic acid or something like that. So here, we're
Right.
seeing
The opposite.
saturated. So it's kind of curious, why do you think there is this whole process of this remodeling if you're only putting concrete back on?
Sure, sure. Well, we have a couple of hypotheses. We're just starting to look into its role in mitochondria now, so forgive me if I overlook anything. But PUFAs are usually more peroxidizable, and you see that in Alzheimer's, where DHA is switched out for palmitate. So this could be playing a protective role in making cardiolipin that's present less peroxidizable and decreasing the amount of
Ensuring that it's more saturated, if in case some unsaturated get in there or something.
Exactly.
And if I understood correctly, it's also expressed in adipose tissue and the heart?
Yes.
What would you think, why would there be, what's the relation? Like, we know
What's
what's
the
the brain, but what's happening in?
We're really interested in knowing that, and we never expected this to be so significant with cardiolipin in brain. We think that it might have a function in just changing the composition of the membrane. So if the membrane is too leaky, CAT2 might be upregulated in order to make it less leaky, but we haven't gone through those pathways yet. We're not sure what it's doing in heart or white adipose.
But do you think if you have a more, if I understood correctly, it favors the saturated fatty
It
acid
does.
distribution? So would you think that would have an effect on inflammatory pathways or something? Sometimes decomposition of phospholipids
Sure.
affect the inflammation.
Right. So you're asking if because
If you
it's
have
the
a
first
higher
saturated.
expression, yeah, would that like render more inflammatory state with the more saturation on the phospholipids?
I'm not sure. I think it might decrease the ability of the mitochondria in energy metabolism because there's usually PUFAs at the SN2 position in cardiolipin. So it might reduce the capacity of the mitochondria to produce energy, but save the mitochondria as a whole if we were to
Thank you. Any other questions?
Hi.
Really nice, Tom. I'm going to get this wrong, but when you express the enzyme that increased cardiolipin in the cell?
Yeah, so we transfected it.
Yeah, okay. So is that surprising to you that there was not increased breakdown too? I mean, I would assume that cardiolipin would try to maintain itself at a steady state level, so
Right.
I'm just curious what you think about that.
I haven't looked into the breakdown, but that's something I'm really interested in looking at. Thank you.
No further question? Thank you, Mark.
Thank you.